so there are two potential problems, as you mentioned. There's off-target effects, other types of unwanted interactions with, say, other proteins, and immune response. So I think the first one is not going to be a huge issue, the specificity, because in all the tests we've done with our design proteins, they tend to be very, very specific for their intended target. And one way to understand that is they're pretty small, certainly compared to antibodies. And so they, and their binding surfaces are really sort of exquisitely designed to bind the target. And we typically don't see interactions with other host proteins. Immunogenicity is another story. Of course, new proteins are foreign. De novo design proteins are foreign. And so how the immune system, in particular the human immune system, will deal with those is an open question. But I can say so far in model animals, we have not seen much immune response. And we think that's because the de novo design proteins are very stable and very soluble. And so they're not likely to be taken up by dendritic cells. And once they're taken up, it's probably more difficult to break them down to present them. And also there are fewer peptides to present. But I think we're going to learn in the next few years from the human clinical trials that are ongoing and will be ongoing, the extent to which humans make a response to them. I think one of the issues about non-canonical amino acids, as I said, they're really great for protein design because they're on the computer, they're made out of atoms and bonds, so you can model them just like you model normal amino acids. And you can also, as I mentioned, extend beyond natural protein backbones to other types of backbone structures. Their manufacturing is a challenge, and so I would add that to the list of the greater difficulty of making compounds that incorporate unnatural amino acid side chains and backbones.